Do you have the last one? This one's on?
I know.
I know, you know what? I was gonna ask you, did you start your study guide?
Yeah, what?
Start your study guide.
Oh, not yet, no.
Because we're gonna start studying like sometime next week. Do you want to study with us?
Yeah.
We usually study upstairs. So what I'll do by Wednesday we'll know our schedule and I'll let you know for sure. Yeah.
If I can, I have an email.
You're what?
Yeah. I just.
I'll send you the stuff.
Okay.
But if I can.
That's all family stuff?
Oh, okay. All right. Okay.
Did you get that?
I'm gonna order them. Black. Take over everything. Now his sweaters are
I would have like.
Well, who sells that? What brand?
Tom Ford.
I never even heard of it.
He used to design for Gucci.
Oh.
He does all the James Bond stuff.
Do you have Gucci stuff? Prada? Louis Vuitton? And you gotta think about it? Coach?
A Coach, like wallet thing. Prada's like, that's for the same price. You just go to LA and find a nice deal. Again, people, there's no labs today, right? This week? Don't forget. Which is kind of sad because I haven't seen my lab classes either.
What do you see is there?
We have to start setting up the problem because that's going to be extra credit.
The kidney, the brain, the heart, and then the lady, I mean, cut it. What's not there, it's like not the gene, what?
Gene what?
I thought it was a gene that has.
Well, there's a cause.
So I have to explain those also?
How are you?
I'm
doing good.
I got a free
Here you go, young man. Okay, good morning, everyone. I know you guys are excited to be here Monday morning. Aren't you? I am. I get my blood work today. Full physical. I'm always apprehensive because they always come up with, oh, you have cancer. You never know. Especially in my family. Average life expectancy for a male in my family is 58.
Dang.
My dad died at 58, my uncle died at 56. The only one that lived the longest was my grandmother. She lived up to 95. But women live a long time in my family, men don't. But you never know. I'm trying to stay healthy as much as I can. But rest assured, I'll be alive on Wednesday when you have your quiz on genetics and chromosomes, of course. All right. So today we're going to talk about. Last week we talked about DNA, right? Who can tell me what DNA is? First of all, what does it look like? DNA looks like a
ladder.
Spiral staircase or a ladder. And of course, I call it, in a book called The Blueprint of Life, I call it hard drive, but you get the general idea. And then we talked about DNA replication. Now, what is the backbone of DNA? Who can tell me?
Sugar and phosphate.
Sugar and phosphate group, and the sugar is deoxyribose. And then you have all these bases, right? Adenine, thymine, guanine, and adenine pairs up with adenine pairs up with and guanine pairs up with. And then, of course, we talked about the story of this discovery and the people that contributed to the story. And you guys create that table like I asked you to do. How many of you did it? Make sure you do it, okay? Make sure. The one who has, you're going to get all 20 questions correct on the exam. right? You just have to know what the person, for example, if I say Hershey and Chase, what did they do? One person, raise your hand. Hershey and Chase.
They built the model?
No. Watson and Kirk. Hershey and Chase. Give you a hint, there was a blender involved.
They created, like,
Go ahead. Almost there. Remember they used radio labels, right? Radioactive phosphorus, radioactive sulfur. They let that do their thing and then they looked where their activity was, inside of the cell or outside the cell. You have a question, young lady? Oh, that's those two, okay? So again, make sure you know those people. Now today we're going to take this journey and we're going to go from DNA to protein. In other words, we're going to talk about what we call the central dogma. The central dogma, right? Sounds like a movie. Central dogma. And the central dogma says genetic information goes from DNA to RNA, from RNA to protein. Now, there's a specific type of RNA, and of course there was the self-replication going on. We talked about this, and you can actually go backwards too. But of course there are specific RNAs. We'll talk about those in detail shortly, okay? So let's take a look at this journey. Of course, the whole point of central dogma is to synthesize proteins. Now, who can tell me what a protein is? Go ahead.
Amino chain.
Chain of amino acids, right? Amino acid. There. Side means killing. Amino killings. You're so violent. You're so violent. Amino acids, okay? Chain of amino acids linked together by a peptide. Bond. I don't know if I have any. Oh yeah, actually I do have some. Let's see if the internet is connected. I hyperlinked stuff last night so I can show you some animation stuff. Hopefully you're still there. So let's see if this works. So I'm going to show you what protein synthesis is all about. So you're going to see the whole picture and then we're going to take it from there. Again, people, while we're waiting, definitely write those people's name down and look at them. So this is the whole process of protein synthesis, all right? I'm going to show you the whole process. If there's any advertising, I do not support it.
Paralyzed by the taste of turkey, but baffled by the burdens of grinding? Join me next week and I'll show you some awesome techniques to make a moist, flavorful, and delicious bird.
People have a lot of free time on their hands, don't they? Jesus Louise. All right, this is more important. Pay attention.
With computer animation, we can enter the cell to view this remarkable system at work.
That's the nuclear board. What is that?
DNA.
After entering the heart of the cell, we see the tightly wound strands of DNA, storehouses for the instructions necessary to build every protein in an organism.
Music is a bit dramatic.
In a process known as transcription, a molecular machine first unwinds a section of the DNA helix to expose the genetic instructions needed to assemble a specific protein molecule. Another machine then copies these instructions to form a molecule known as messenger RNA. When transcription is complete, the slender RNA strand carries the genetic information through the nuclear pore complex, the gatekeeper for traffic in and out of the cell nucleus. The messenger RNA strand is directed to a two-part molecular factory called a ribosome. After attaching itself securely, the process of translation begins.
talk about these key players.
Inside the ribosome, a molecular assembly line builds a specifically sequenced chain of amino acids. These amino acids are transported from other parts of the cell and then linked into chains often hundreds of units long. Their sequential arrangement determines the type of protein manufactured.
At the bottom of the chain.
When the chain is finished, it is moved from the ribosome to a barrel-shaped machine that helps fold it into the precise shape critical to its function.
Waiting for a superhero to jump in.
After the chain is folded into a protein, it is then released and shepherded by another molecular machine to the exact location where it is needed.
So it's a fascinating journey, and it's an extremely important journey, and we're going to talk about it, all right? That's what they were talking about. DNA going to, now to be specific, we're going to what we call messenger RNA, and then from messenger RNA you go to protein. This part right here is called transcription. And it takes place in the cytoplasm, in the nucleus. It takes place inside of the nucleus. While going from RNA to protein, this part is called translation. And this takes place in the cytoplasm. So on the exam, if I ask you, going from DNA to messenger RNA or RNA, what do we call that? Your response should be? Transcription. Going from messenger RNA to protein, we call that? Translation. And where do they take place? Your response should be in the? Nucleus and the cytoplasm, right? Right there. 4, 5 different test questions right there on the spot for you. Okay? All right, so let's continue on. So the process of translating, in other words, the process of getting proteins involved, 2 steps: transcription and translation. Transcription and translation. And you're going to learn today how transcription and translation take place, albeit in a very abbreviated version. All right? Abbreviated version. For me to cover this stuff, it will probably take me about 3 weeks, 4 weeks to cover it in a lot of detail. There's lots of details involved. All right? But nevertheless, it's still a fascinating journey, so let's take a look at it, all right? So this is the overview of the lecture. We'll transcribe DNA into messenger RNA. Messenger RNA, which occurs in the nucleus, will move to the cytoplasm, and there it gets translated into a peptide chain, which is nothing more than a chain of amino acids, all right? And of course, ribosomes are involved in that process. So the central dogma states, again, DNA goes to RNA, RNA goes to protein. DNA goes to RNA, RNA goes to protein. Now, we're already talking about DNA in detail. And I already have introduced RNA to you guys way when we covered the molecular stuff, all its bases and so forth. But we're going to talk about RNA in a little bit more detail today, too. All right, we're going to talk about a little bit more detail. First of all, so let's take a look at RNAs. There are 3 different types of RNAs that you need to be aware of. There are 3 different types of RNAs. Don't copy everything on the board because it is in your PowerPoint, right? Now, messenger RNA, it's in the name, EA4. What do messengers do? Carries a message, right? So the message of making the protein is in the messenger RNA. The message of making the protein is in the where? Messenger RNA. Ribosomal RNA, I want you to write this down, ribosomal RNA, also known as rRNA, by the way. So messenger RNA is mRNA, ribosomal RNA is called rRNA, R lowercase. rRNA is like the workbench. rRNA is the workbench. What do you do on a workbench? Build stuff, right? Now, go back to that video. Remember that messenger RNA that came out of the nucleus? And it went into those ribosomes, right? That's the ribosomal RNA there, okay? and it sits there, so you can build stuff on it, all right? And then, of course, you have the transfer RNA. The transfer RNA, and I want you to write this down, transfer RNA is the workhorse of this whole process. Transfer RNA, or tRNA for short, is the workhorse. So transfer RNA is the what? Now, in that video, some of those little things that are coming in and putting pieces together, those are the transfer RNAs. Those are the transfer RNAs. By the way, how do you make messenger RNA? How do you make ribosomal RNA? How do you make transfer RNA? You make all of that from DNA. You make that from the DNA. Different parts of the DNA will code for it. And of course, there are proteins involved too. So this is that whole chicken and egg scenario, right? Which one came first? Well, in order to make RNA, you need DNA. But in order to do that, you also need protein. So people used to say, well, how the hell that is happening? Well, we know for now that the first genetic information were RNA. Because RNA not only can self-replicate, but can only produce protein, but can also act as an enzyme, and it can go back and make DNA. So the first molecules that evolved most likely were RNA, and after all, take a look at it. RNA is a single strand. RNA is a single strand. So, what are some of the characteristics of RNA? First of all, the backbone of RNA is just like DNA. The backbone is just like DNA, so pay attention here. You have the phosphate group. You have the phosphate group, and then you have a sugar. But notice the sugar. What is the sugar here now? DNA, the sugar was? Deoxyribose. And the difference is this oxygen right here. Difference is this oxygen right here. So the backbone is the same. And of course they have the same basis except instead of thymine, RNA does not have thymine in it. RNA does not have thymine in it. Instead of thymine, it has uracil. RNA, instead of thymine, it has what? Uracil. Furthermore, these are all characteristics of RNA. You need to know this. Furthermore, RNA is single-stranded. Specific, I should say messenger RNA. Furthermore, messenger RNA is single-stranded. It is shorter than the DNA, and it is shorter than the DNA. If you pay attention to that video, notice only a small section of the DNA unwinded itself, didn't it? Not the whole DNA. And you only translated or transcribed that one section. That's why RNAs tend to be shorter. So imagine you have a book of spies and that codes in. You only read a certain part of it. Okay? Even within that, there's codes. Any questions so far? Now, RNA base pairs with a strand of DNA. So imagine if I have a strand of DNA. Let me turn this up so you can pay attention to me. This is a DNA, okay? You open up a strand, so imagine there's more here, but you open up a strand. You can use either one of these two to build the messenger RNA, right? You can use either this one or this one. And by the way, the way you put the nucleotide is just like DNA replication, 5' to 3' direction. So that's how base pairing takes place. Now, just like DNA, the RNA, if you have a cytosine on DNA, you get a guanine on RNA. If you have a guanine, you get a cytosine, all right? But if you have a thymine, it base pairs with adenine and RNA. But if you have an adenine, since RNA doesn't have thymine, it base paired with uracil. And I'm going to do that on the board back there. So,
Back there?
yes, sorry people. Don't blame me. Hate the crackhead designer of this classroom. All right? I swear. I still wonder where they built a better room than this one. So imagine you have a DNA strand. We find TA, everything starts with a top box anyway. Guanine, cytosine, guanine, adenine, cytosine, so imagine you have a strand of DNA. It's only one strand, necessarily not. Imagine you have separated the strands. So on the exam, I'll just give you one strand, so it's easy. You don't have to work the first one I do, okay? So your messenger RNA will build from a 5' to 3' direction, right? So the thymine will pair up with adenine. But adenine will pair up with uracil. Thymine will pair up with adenine. Adenine will pair up with cytosine pairs up with. Guanine pairs up with. Cytosine pairs up with. Cytosine pairs up with. What is this one? Uracil and uracil acid. Right? Now you have your messenger RNA, right? I shouldn't do these lines. Then this messenger RNA, this is the nucleus. That's the nucleopore. This messenger RNA will come in and so on the exam, if I give you a strand, you want to know if it's an RNA or a DNA, look for uracil, right? If there's uracil in it, we're talking about RNA. And I won't be tricky and give you a message. Technically, you can have a messenger RNA with no uracil in it, but I won't be that tricky. All right, so everybody comfortable with this? Can I be flip again?
Horrible, bro. Horrible.
All right. So again, just like DNA replication, nucleotides are added from 5' to 3' direction. But unlike DNA, only a small stretch is at the template. You saw that. Only a small part unwinds itself. Only a small part unwinds itself. Now the enzyme that unwinds it. Remember in DNA replication you had DNA polymerase? Now you have DNA RNA. The one that was unwinding it, that is called RNA polymerase. Okay, so make sure you know that molecule is extremely important. And of course, again, product is a single short messenger RNA that is ready to leave the nucleus. Pretty straightforward, right? Pretty straightforward. Now this is where it gets complicated. I actually took this from my notes when I took this class. And we covered this in 2 weeks, and my head was spinning, and it's still spinning every time I read my notes. One, my notes were horrible. But it is a very complicated process. It's well understood, but it's not in my field. Dr. Rainbow can probably talk about this for years. How many of you have Dr. Rainbow for lab? No. All right. He will cover this with his eyes closed and his hand tied behind his back. All right? So that's his specialty. He knows this stuff more than anyone on this campus. All right? So what happens is you have a site called a promoter. Promoters are important because the question is, where do you start, right? How does the RNA polymerase know where to start this unwinding process and so forth? Well, notice at the beginning I put TATA. That's called a Tata box. That is called a Tata box. Think of the Tata box as the switch. All right? And the promoter is the one who turns the switch on. All right? So you have a base sequence DNA that is signals the start of the gene. That tells the DNA this is where the gene is starting. Because your genome is a junk. Quite a bit of it is a junk. Right? Quite a bit of it is junk. Stuff that doesn't code for anything. Some of it have support functions. Some of it could have had a function, but it got mutated out. A lot of it, not a lot of it, but a good chunk of it is actually endogenous retroviruses. These are the viruses that have inserted their DNA in your body. So we walking around carrying virus DNA, all right? So it's essential for the cell to recognize which site it's supposed to sit on, and that's where the promoter comes in. So for transcription to occur, the RNA polymerase must first bind to the promoter. And then you get a sequence of events. So RNA polymerase will bind, all right? And then you get what we call these transcription factors. They come in and they assemble themselves. Once transcription factors come in, and then you get this activation transcription factor that comes in and kickstarts the process. And you have this ATF binding site that will sit on. And then you got that you assemble that weird little creature-looking thing that was doing this. That's how you assemble it, all right? Now this is a highly abbreviated version. If you find this stuff fascinating, which I do, how many of you are going to take Bio 110? I think you are for sure. You guys are going to spend 2 weeks on this, so. All right? So this is where the promoter is, and then of course you have the gene. So if you get a mutation that disrupts the promoter, what's going to happen to the gene? Are you going to make the gene? You're not going to read the gene. It's over, right? and these mutations could use any. So once everything is assembled, the RNA polymerase, it just goes along, and then you start building the nucleotides like you guys did. And I will make you guys do one on the exam, all right? I'll have you build one on the exam, yes.
A promoter is always TATA?
That's one of them, but for the purpose of this class, yes. Okay. And of course, sequence are at. Again, is everybody comfortable building the sequence? If you can build the DNA, you can build this one. The most common mistake I see is students, instead of putting uracil, they put thymine. So just remember, RNA, uracil. Okay? RNA builds what? Uracil. All right. Young man, are you sleepy? Go home. Keep your eyes open. Next time you kick you out. Now, when you transcribe a section of an RNA, section of a DNA, when you transcribe a section of DNA, again, not all of it is the stuff that's readable. Some of the stuff is actually junk. The stuff that are important, we call them exons. The stuff that is not important, we call them introns. So initially, when you transcribe your RNA, you transcribe the whole thing. And you put caps on it to protect it. But you transcribe both the exons and the introns. So imagine you are translating a book. There's a book I wrote a book in English, and you want to translate it, let's say, into Spanish. But when I wrote that book, I wrote, in this book, and then I put gibberish, 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 gibberish. I will be gibberish, 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 talking about gibberish, gibberish, DNA transcription. And you're going to translate that to Spanish, all right? Now, I'm not going to even attempt to do that, because I'll probably butcher it. How do you say gibberish in Spanish? Gibberishia, all right? Now, initially, the translator will translate the whole section. The good words and all the... And then the enzyme comes in. The enzyme comes in and starts snipping out the introns. These enzymes, they bind to a specific area, so they know that's it. They bind to it and they'll just snip it out. Take those out and eventually get recycled. And then you put the exons together. So notice, the messenger RNA that actually leaves the nucleus is much shorter than the whole section that you transcribe. Same thing in that book. Once he or she translates that book, they go in there and then they just take out the good parts and then get rid of the gibberish parts. All right? Yes?
What did you say cuts out the introns?
Enzymes. All right? Enzymes. And we'll talk about these enzymes when we talk about gene technology. These are the enzymes we use to slice out DNA and insert them into other animals. All right, something that we can do in this school right here. Now, I don't know if I told you guys, but we are reviving our biotechnology class. And in that, you're going to learn how to isolate DNA, do genetic fingerprinting, it's like CSI kind of stuff, and cut DNA and do other stuff. All right? Any questions so far? Now, let's talk about the genetic code. Now, give you a short break and come back. And I apologize, but I have to make you guys turn around again. Yes?
Do we have to know that, we do not
Yes.
have to know the exon is important and that the intron is just gibberish and at the end that it turns into a short RNA strand, right?
Yes.
Pretty much that's how you
What?
cut off the enzyme.
DNA splicing? Splicing, that's something else. We'll do that next week. All right, so pay attention here, people. This is where it gets tight. And I want to make sure you guys get this. But if you get this on the exam, you're going to do well. By the way, I am working on getting a room to do our review session for the exam. But if I can't, I'll just do the review in class like I did last time. Okay? And I'm also going to give you a little bit more in-class guiding. Remember, the test is comprehensive. After this, from last test on, I'll guide you a little bit else for the other parts, just so you know what to expect. Okay? Now, everybody clear about this part, right? So I'm going to take out the DNA and only leave the RNA. No need for confusion. I know I'm probably going to come back to it, but take this out. Take the snake out. All right. Now, when it comes to taking this messenger RNA and translating it into protein, which is amino acids, You read it in sequence of 3. How many of you are familiar with computers here? What's the language, a binary language of computers? Zeros and one, right? Zeros and one. Here we use 3 codes. So every 3 codes are specific for something. Every 3 are specific for a specific, acid. Then we have ABAC. Each one of these is called a codon. So every triplicate is called a what? Codon. Every triplicate is called a codon. Now, RNA has how many bases in it? 4, right? Adenine, uracil, guanine, and cytosine, right?
Mm-hmm.
So, my question to you, my dear students, how many codons are possible? There's a formula too. Every three is a codon, right? How many bases do we have?
4.
So 3 to the 4th power, I'm sorry,
3 to the 4.
4 to the 3rd power,
64.
it is 60. Therefore, there are 64 possible codons. Everybody with me? Can I get a help with that? Now, out of these 64 possible codons, 3 of them are stop codons, which do not code for any amino acid. So out of 64, how many we have? Stop? 3. Now, those are the ones you need to memorize. I'll tell you how to memorize them. But 3 of them are stop codon, and they don't code for anything. So on the exam, if I ask you, what is a stop codon? You say it's a codon that is not coding for any amino acid. It's a codon that doesn't code for any amino acid. Now, the start codon, that codes for amino acids methionine. That's always the only start you have, all right? Now, this is where it gets interesting. So that leaves me with 61 possible codons, right? 61 possible codons. All right, let's ask you, Jack Sickley. That's what happens when you become volunteer to answer the question. How many amino acids are out there in nature? How many amino acids are out there? 20. Right? We have 20. What gave it away? Okay, this is important. Let's pay attention. There are 61 possible codons, right? And each codon will code for an amino acid. The codon codes for an amino acid, okay? Codon codes for an amino acid. The codon codes for an amino acid. Everybody with me? So if there are 61 codons, but 20 amino acids, what is that telling me? There's 61 codons, but 20 amino acids. What is that telling you? That means there is redundancy in the genetic codes. And I think that's what he's trying to say when he's moving the 10. Okay? There is redundancy in the genetic code. You can have more than one codon to code for a specific amino acid. So imagine that book is written, the letter "the" can have 6 different meanings. That gets confusing. That's why I laugh when they say human genome is design. If it's design, why do we have gibberish in it, first of all? And not only why do we have gibberish, why so much redundancy? All right, so again, turn around now. There is redundancy in the genetic code. There is redundancy in the genetic code. So, 64 codons, 61 specify amino acid, 3 specify stop. And the stop is nothing more than a lack of amino acids, right? So there is redundancy here. Amino acid leucine can be coded by uracil, uracil adenine, uracil, uracil guanine, cytosine, uracil, uracil, cytosine, uracil, cytosine, cytosine, uracil, adenine, cytosine, uracil, adenine. Yes, in hard order. My night class, I was so tired by environmental. I'm lunching and I'm pointing to this here. And I'm like, you guys see it? They're like, no. I'm like, oh, okay. All right, any questions so far? This is what the code looks like. And this code is universal. This code is almost universal. Very few exceptions are found. The biggest exception is in prokaryotes, instead of methionine for the start, you have S-methionine. Instead of methionine, you have S-methionine. Now, do you have to memorize this? Absolutely not. I will give you this. But you have to learn how to read it. Okay? And that's right after the break. Go ahead and take a, I feel generous, 4-minute break. Yes, no need for father.
And also like the other one, the third one is so 3 will be 1.5.
Wow, some people dropped off the face of the map. They're what?
I noticed that too. I didn't know why.
We all scored ours one way or another. Unless you're the Mexican clan.
I can't believe that.
Hilarious.
So if I Google it, it'll come up?
I stumbled on it by accident. I was researching like the concept of race in biology and stuff like that. I'm trying to put a talk together that why in biology race makes no sense. It's just a man-made thing.
Right.
And I stumbled on that.
Ethnicity, not race.
What?
It's ethnicity. Race is like a human race.
Yeah, no, ethnicity is different than race. But race is something that's race. There is no race. There is no Caucasian. That doesn't exist.
Are we gonna talk about
There was a cycling, I clicked on it.
Are we gonna talk about like the human tree and stuff? Like how like it starts from Africa and the migration?
All the
I just remember the last one. Oh, that's all. But then the other two.
All right, people, let's finish up. Maybe I have time to do more examples. So, pay attention. So when we went from DNA to messenger RNA, we call that transcription, right? It occurs in the nucleus. It's not that hard, right? It's not that hard. And you should be able to transcribe. But now we're going to talk about translation. This is where it gets kind of dicey because there's a lot of players involved. This is how translation works, okay? So imagine you have a sequence. And I think you guys wrote it down, the one I wrote on the board, right? So the first codon is what? AUA, right? The first codon, the one you wrote down is AUA. So this is what you're going to do. You're going to say, well, the first codon is A. You see that? The second codon is U, and then the third codon is A. So you triangulate, and you end up with AUA. So AUA will code with which amino acid? Isoleucine, right? Isoleucine. Now, technically, if that was the right sequence I've written, that was the promoter. Promoter doesn't become part of that. It's not part of the promoter. The actual one is AUG. That's where the start is. All of them start with methionine, right? So on the exam, if I give you a strand of RNA and have you start the process of putting DNAs together, always start with AUG. And I think all the examples I've given you, it starts with AUG anyway, so you don't have to worry about it, all right? So let's look at the second sequence. Second sequence is UCG, right? The examples we did. So, U, C, G, so UCG codes for serine. See how that works? The third one, GCU. You have G, C, U, GCU, alanine. See how that works? So go ahead and do the UGG and GAU on that code that you did. Just do the last two codons, please. Quietly, without, you can look at the board, you can look at your notes. Just do it quietly, please, because you have to be able to do this on the exam. Por favore. That means now.
Mhm.
10 more seconds. Nine Mississippi, 1 Tennessee. Hands up. All right, so second, so is UGG, right? So what is UGG code for?
Tryptophan.
Well, let's look at
Fine.
it. UGG. Tryptophan. By the way, this Sunday, you guys gonna be eating, this weekend or Thursday, you're gonna be eating a lot of tryptophan. Turkey is loaded with tryptophan. And guess what tryptophan makes you?
Maybe.
Sleepy.
Is that why they have you ever heard of the itis? Yeah. Like when you eat a lot and then after you eat, you're like really tired?
Here's an idea, don't eat a lot. Think of, when you guys gorging yourself on that turkey, imagine those people who don't have food, that they eat one meal a day.
So when you do it, you do the first letter,
Okay, pay attention.
you do the first letter, which is U,
Okay?
and then you do the second letter, which is
G.
G, and then you look on the side for the third letter.
And then you just bring them together.
Okay.
G.
Oh, okay.
All right, GAU. So, what was that coding for? So you have G, A, U. Is that hard? And again, this code, I don't care if you're an insect, a fungi, heck, even a politician. The same language reads. With the exception, the one I know of is bacteria. Instead of methionine, you have ethmethine. Now, the only codons that I want you to memorize are the stop codons, which are UAA, UAG, UGA. These are called stop codons. UAA, UAG, and UGA. And what is a stop codon? Stop codon is a lack of amino acid. Make sure you write that down. Stop codon is a lack of amino acid. You have a question?
Yeah, so the promoter is the start codon?
No, the promoter is actually before the stop codon. That's all around here, okay? Promoter builds up, and then the first amino acid sequence is UAG, and then that becomes methionine, all right? This is the start in terms of making the protein, not the transcription. This is the transcription. Now, this is important. This is where students get confused, and sometimes they panic on the exam. You'll get a strand yay big, okay? And you start doing the transcription and translation, right? So you have the DNA, and then you translate it. Don't worry about introns and exons. I'm going to just assume it's all exons. But halfway in that strand, I will insert one of these codes, UAA, UAG, and UGA. Once you hit that codon, you stop. Once you hit that codon, you stop. So your strand might be this long. If you hit that codon, that's all you have to do on the exam. Okay? That's all you have to do. All right? Now in nature, usually stop codon is the last one, but sometimes you get a mutation. Notice, to go from, right here, tyrosine, all you need is a mutation to change this C to an A or a G. Now you have a premature stop. And a premature stop will make the protein to be much shorter. All right? Make the protein shorter. So on the exam, once you hit the